Okay. So my protein was tubulin.
Gotta talk loud.
My protein was tubulin, and my name is Joshua Fleming. So the first part, I wanted to explain exactly what a protein was. And so I have like different types of proteins, which include enzymes, structural proteins, transport proteins, motor proteins, storage proteins, and signal proteins, and that's just to name a few. There's so many out there. And then for functions of proteins, I put so proteins can help with catalyzing covalent bonds. That's the enzymes, and which is like breaking and forming the bonds as far as catalyzing goes. And they provide support for tissues, carry small molecules and ions across like the membrane as like transport proteins. They can store amino acids and even generate movement within the cell. They're very important to the cell. And so there's 4 levels of a protein. There's primary level, which is like the unique amino acid sequence. There's secondary level of structure, which is your alpha helices and your beta sheets. And then there's tertiary level, which is bonds between the R groups. And then there's quaternary level, which is the bonding between 2 or more like structurally functional proteins. So these are just like the levels. You can see here, these are your alpha helices, they're swirlies. And then beta sheets, which are like straight arrows. Tertiary is once it starts folding on itself and bonding and stuff. And then your quaternary is going to be a bunch of them put together. So the function of tubulin, the main function is they make up microtubules, which is like structural support for the cell. They're important in mitosis of the cell, in eukaryotic cytoskeletons, which is like referring back to the microtubules. Intracellular support, which refers also back to the microtubule. Catalyzing, which is like there's an enzyme tubulin out there. Intracellular transport, so like the molecules go through the tubules. And then, so next I have like your types. So your tubulin GTP phase is going to be your enzyme tubulin. Your Y tubulin, beta tubulin, and alpha tubulin make up the microtubules. And here's like a microtubule. You see your alpha and beta tubulin, they bind together to form that quaternary structure as a microtubule. Tubulin structures, so like on average, a tubulin structure has about 500 amino acids, which is pretty complex since there's only 20 different amino acids out there. The primary structure of tubulin is like the amino acid sequence, so like the different arrangement of the 20 different amino acids adding up to 500. And then the secondary structures of tubulin are like the alpha helices, which are like there's a lot of them out there. There's like 40% made up of alpha helices, and those are for like the transporting systems. And then there's the tertiary structures, which is the bonding in the protein when it folds back on itself. And then the quaternary structures, which rely heavily on the van der Waals forces to bond 2 of the proteins together. So this is like a protein structure of tubulin. You've got your helices, your sheets, your tertiary structures, and then your quaternary structures. And then tubulin, DNA, and gene coding. So in research that I found, there's been a lot of like studying with alpha and beta coding. As far as like mutations go, there's all kinds of things like Joubert syndrome, optic nerve damage, Alzheimer's, hematologic malignancy, and Down syndrome. And so like researchers are trying to figure out how like they can change the DNA sequencing to make better tubulin so these won't exist anymore. Gene coding is like, so you've got your gene, which is like a segment of DNA, and it is like the coding for tubulin through transcription and translation. And that's it for my tubulin PowerPoint essay.